And Karin's going to talk to us today about text binding improves prediction of protein functional sites. We look forward to your remarks.
Thank you. Hi, everybody. I stand between you and lunch, so I will try to make it interesting. So I'm going to be talking to you today about some work that I've been doing in collaboration with Michael Wall and Judith Cohn at Los Alamos National Laboratory in order to try to show how we can use text mining in combination with a structural prediction model for trying to improve our capacity to do high throughput annotation of protein functional sites. So why do we care about this problem? I think most people in this room probably appreciate this already, but I'll just say it to provide a little context for our work. And the basic idea is that, well, we have the blueprints to life, we really don't know how to read them. So we really need to move beyond just having a genome sequence to understanding how to modulate and manipulate proteins in order to ultimately treat diseases that we are concerned about. So we're really interested in trying to identify the sites on a protein that control the behavior of that protein. So that's really the ultimate goal of what we're trying to accomplish. The way that we're approaching that is, as I said, to integrate two different models. Essentially, we're doing two different processing of two different information sources in parallel and then bringing them together in order to enable a very high throughput approach to functional site prediction that also has high confidence and evidence from the literature in order to support that. So we're integrating the structural modeling with text mining that's actually searching for validating evidence. So just to provide another bit of context, there is a lot of work about protein function that really targets specific vocabularies like the gene ontology. So protein function in a lot of work is defined by these concepts in that controlled vocabulary. What we're talking about here is really a physical definition of protein function. So really looking for specific sites on the surface of a protein that are ultimately responsible for that protein's behavior. And these can be catalytic sites, ligand binding sites, protein-protein interaction sites, and so on. Now, the starting point here is that, again, we have the blueprints to life. That's in the form of a two-dimensional sequence. And the key issue for functional site prediction is that functional residues are often quite remote in sequence. So I've highlighted in red a couple of residues in this sequence which together form a catalytic triad. So if you look at the three-dimensional structure for that protein, while those residues are quite far apart and no clear indication that they interact in any way or that they're related in any way in the two-dimensional structure, once we put it into a three-dimensional context, we can see that they're actually quite close together in a physical sense. So this motivates our approach in looking at the three-dimensional structure. So my colleague, Mike Wall, and Judith Cohn from Los Alamos have developed a method called dynamics perturbation analysis, which tries to identify, based on the physical properties of the protein structure, the sites on that protein that are functionally active. So their basic method, and I won't spend a lot of time on it, but just to give you an idea, is to color the surface of the protein with points, and then to try to, essentially they tickle the protein at all of those points, and they measure the conformational change of the protein at those points. And once they've done that, they look for the sites that have the most conformational change in that process, and they predict those the residues at those sites to be the functionally active sites in the protein. And so this method has been shown in previous work, which I cited on the last slide, to actually be quite accurate. So it's doing a pretty good job in general of finding actually functionally important residues. So on the on your right, I guess, we have the predicted residues from the DPA methodology, and on the left we have the actual catalytic triad that has been verified experimentally. Okay, so as I said, we're going to try to bring text mining into the process here, and our strategy is to take advantage of information in the PDB for the protein structure, in order to search the literature effectively for evidence supporting the prediction. We're going to take an extremely simple hypothesis. So I don't want to give you any idea that we're actually doing very, very sophisticated text mining here. We're not. In fact, we started with the idea that we should keep it simple, stupid, and just assume that any mention of a specific residue from a protein in a related publication is meaningful. So the idea is that an author who's writing up a result is not going to mention a specific site on a specific protein unless there's some functional reason to do so. And the rest of the talk is basically to convince you that that's actually true. So as I said, what we've done is take advantage of the information in the PDB to drive both the structural processing and the text mining. So the structural information, the 3D atomic information in the PDB is used to drive the dynamics perturbation analysis, while the primary citation of that record, which points out to a PubMed abstract, is used to pull text in that's information that's relevant to that protein. Then we apply our very simple text mining pipeline and pull out the residues that are mentioned in that abstract. And ultimately, we bring these two things back together in order to accomplish two things. Essentially, to constrain any errors in the text mining, we can go back to the original sequence and validate physically the residues that we've pulled out of the text, and then also to have this amplifying effect of finding evidence in the literature to support our structural predictions. And so we call this a kind of validation process. We're using the literature to validate the predictions of the structural model. So the text mining system itself is a set of very simple patterns that recognize amino acids located at specific regions in the text. We're taking advantage here of the regularity of expression of residue mentions. So there's a limited vocabulary. There's only 20 amino acids, and then we have the positions within the two-dimensional structure that are referred to. Again, we're relating the specific abstracts to a protein using the PDB primary reference, so this means we can avoid having to do an open-ended information retrieval for all the information about a specific protein. And that's a limitation, but at the moment we're using that to really make sure that the information we're pulling in is relevant to the protein. And as a proof of concept, that's clearly something we need to show it works in the limited case before we try to open it up to the noise of the more general case. So that's what we're doing here. As I said, we're using the sequence information from the PDB to filter residue mentions that don't match a residue in the protein. So some of the examples of the kinds of things that we're pulling out with our patterns are the typical things you would expect. So the the three-letter abbreviations along with a position number. But we're also handling some more linguistic variation in how the information can be expressed. So we're looking for phrases like substitute serine for proline at position 97. We deal with some surface variation, like sometimes there are hyphens and sometimes there are knots. Lists cysteines at position 6, 42, 48, 90, and 393 were replaced by serine, and single letter abbreviations as well. So we try to handle all of these cases. Of course, we have to assess how well our system does on these, and what we have here minus and plus on single letter amino acid patterns and single letter mutation patterns, because the single letter variants of expression are highly ambiguous, and so we wanted to see whether or not we should include them. And you can see the results here. Basically, the results reflect the regularity of the patterns that you would expect. There are other systems out there that deal specifically with finding mutations, so we're able to test our mutation patterns on the corpora that are available. There are very few resources for just bare amino acid residue mentions. There is one that I show here, the Nagel corpus, and we also developed a full text corpus to try to assess this as well. The performance on full text drops from what you see in the abstracts, and that's because there's more variability in how sites are referred to there. All of the corpora that I've referred to are available online at that URL. So what we saw actually in these results is that if we include at the very last line, system 4, if we include the single letter abbreviations for both the amino acids and the mutations, we get a very high recall, and our precision drops correspondingly because of the ambiguity that I mentioned. But since we're in the context of using the physical information of the protein to constrain our residue detection, we can actually go with the highest recall system and let the physical structure essentially filter out the false positives that are contributing to the lower precision there. So that's what we do. We've applied this strategy to approximately 100,000 protein structures, domains that are in the PDB. We've done DPA analysis on all of them, and we've done text mining over the associated primary references where they were available. In the results that I'm reporting, I'm focusing on abstracts. We have also run this on full text, but I won't talk about those results here because we haven't analyzed them sufficiently yet. So the basic idea is that we take a PDB entry, we pull out the primary reference, we identify all the text mentions or text occurrences of the raw amino acids and the mutation sites, and we normalize those in order to compensate for the linguistic variation that we see, and then we map them back to the physical residues in the protein sequence. And what we're seeing in this particular example is that we've pulled out arginine 33, and it actually maps back to 2 different chains within the PDB structure here. So there is some ambiguity even in that physical matching step. The basic results of the approach that are worked through in the paper in a lot of detail are that DPA by itself can preferentially highlight functionally relevant sites compared to all other sites. And in this case, we're looking at information, again, from the PDB itself. So near small molecule sites are basically sites on the protein structure that have a small molecule embedded into the structure when the structure was crystallized. And those are considered to be functionally important sites because usually the protein won't crystallize properly unless that small molecule or the ligand is there in situ. On the residues that we identified through the text processing, we showed are 6 times more likely to be found in a known functional site than other residues. So 39% of the residues that we pulled out of the text processing matched a curated near small molecule site. So these are sites coming out of something called the binding mode database, which catalogs protein ligand binding sites, or the catalytic site atlas, which captures catalytic sites. And only 6% of residues without a text mention matched one of those annotations. And when we bring the 2 systems together, we find that the overlap of predictions with the curated data sources we have access to improves when we bring them together. So just to give you a sense of the coverage of the method, we find over 17,000 abstracts linked to the PDB records we processed. About 6,000 of those we were able to find mention of a specific text residue in the abstract. Remember, these are results from abstracts alone. And 5,236 of those actually had a text residue that we were able to physically verify. So we were able to map those back to the protein sequence. Then we looked at the curated data sources to kind of capture, to get a sense of what information we were recovering. We can't directly calculate precision recall here because we don't for many of the predictions, we don't have curated information. So we can just look at the coverage of the curated sources, and the rest are essentially predictions for where there are functional sites that haven't made it into those curated data sources. So what we see here in green, yellow, and red, the green are direct annotations, so residues that match directly to a particular physical residue in the actual PDB sequence. We did multiple sequence alignment to then generalize that residue match to the protein level. So in the PDB, because PDB is structured at the domain level, you'll have multiple PDB records that correspond to the same protein. So the yellow represents that generalization, and then you have proteins at who are in the same protein family, and again, we do a multiple sequence alignment to match to those residues. When we look at, when we do an analysis of the residue-wise performance of predictions, we're trying to establish how much the text mining is adding. And this might be slightly confusing, but one thing that I probably haven't explained carefully enough is that DPA predictions predict a number of residues as being the functional site. So it's not there's not a one-to-one mapping between sites and residues. But the text residues are picking out the text mining is picking out individual residues. So what this chart is actually looking at is essentially how much of all the residues in a known functional site are we recovering with the different methods. And the thing to probably focus on is what happens at the recall of 0.5 or precision of 0.5. And if you just look at the top, if you look at the sort of dotted blue line, light blue line, that's contrast that with the red line, essentially that's the benefit we're getting from adding in the text mining over the strict DPA approach. So we're seeing a big bump in our quality by adding in the text mining along with the structural prediction. So just to give you one concrete example of what this looks like, this is a protein from the PDB, 1YK3, and what you see in yellow is the DPA prediction for the active site, and what you see in orange and magenta are specific residues that were identified from the text mining. And of course, I'm going to cherry pick a nice example to show you, but what you see here quite clearly is that the residues we've picked out with the text mining are right in the middle of that site. So we were quite happy with this. We decided to go a bit further and actually look at 10 PDB structures for which we had predictions that were not in our curated data sources to kind of get a sense of how accurate our text mining based predictions were. And what we found, I don't expect you to read that, the table's in the paper so you can look at it in more detail, but the basic point here is that we went out to the literature for each of those 10 structures, and the language in the literature supported the idea that those were actually functionally important sites. Another way to look at this information is to look at the conservation of the residues that we're picking out with the text mining as compared to other residues. And because we know that the evolutionary pressures on residues in protein functional interfaces are different from other residues. So we expect residues in those functional interfaces to be more highly conserved. And as it turns out, the residues that we find in the text are on average more conserved than the other residues that are not mentioned in the text. So again, this is evidence that what we're picking out with the text mining are functionally important residues. So that essentially is the approach, and the implications of the approach or the conclusions there are that we can utilize simple text mining techniques to increase our confidence in structurally based protein active site predictions, and we believe that these high confidence predictions are strong drug target candidates. And what we've produced then is a method that has high throughput literature supported protein function prediction. So we're able, as I said, to do this for all of the proteins in the PDB and provide high confidence predictions for those. We are in the process of putting this up on a website, which is not quite available yet, but this is a screenshot of what it will probably look like. We've juxtaposed the text abstracts that we've processed alongside of the actual three-dimensional structures of the protein. That window, obviously here is a static screenshot, but it's an active JMOL window, so you can actually manipulate the protein structure. We've highlighted on top of it the predictions that we found, both from DPA and from the text mining. So the blue residue, which is a little hard to see, it's up there on the corner, is a residue that was mentioned only in the text. The red ones are predictions in the DPA site, residues in the DPA site, and the magenta are residues where the two overlap. And the color on the abstract that overlays matches those things. So it should provide a nice way for people who are interested in exploring these functional sites to actually look at it in context, providing both the three-dimensional context, but also the abstract where those specific residues are discussed. As I said, we think this is a good starting point for drug discovery. This is just a pitch right now. It's not actually validated, so we're hoping to find somebody who actually wants to try to use this in a particular drug discovery context. But the idea is that once you've identified the functional sites on the protein, you can either do structure-based drug design by taking advantage of that structure, or do virtual drug screening in a way that prioritizes prioritizes where to target the drugs using the predictions of our integrated model. I mentioned several limitations, and clearly we are wanting to now that we've validated the hypothesis that the text mining does add value to this process, we can start trying to generalize the approach to handle more than just structures in the PDB, but actually to try to work with predicted structures and to try to generalize to proteins outside of what is available in the PDB. We also want to look at other active site types. So the DPA model right now is pretty focused on catalytic site and protein binding site, but we also wanted to start looking at protein-protein interactions. And of course, we want to extend to a much broader set of literature. I'll just mention, this came out badly, sorry, I don't know, Mac to PC conversion problem, but this is obviously too much detail for me to get into, but one step in the direction of trying to generalize our text mining methodology is to try to establish protein amino acid relationships directly in the text so that we can take advantage of that broader set of literature and not just rely on the curated link to the literature from the PDB. And so this is just a pointer to a paper that we have in press at the moment, which is about learning those relationships in text. And we get quite high performance on that specific information extraction task, and I'm sorry that got all messed up. And in order to do that, we actually used our physical validation process to bootstrap a corpus of training examples, because anybody who's ever done anything with text mining knows that half the battle is getting training data for your algorithms. And so we came up with a methodology based on the distant learning paradigm and the use of the physical information that we have in the PDB to generate training examples for this purpose. So in sum, I wanted to introduce you to this methodology and just point out that the architecture of the approach that we've defined is essentially a new way of using text mining, because we're really thinking about using the text mining here as this validation step to validate the predictions of this orthogonal structure-based model. And by using the biological context, we can really constrain and improve the literature mining as well. So it's a tightly coupled approach that takes advantage of the best of both worlds in some sense and takes advantage of the full context of the information. And I will stop there and say thank you very much and acknowledge money.
Hi. So, if I understand correctly, you're looking mostly at abstracts of articles, but for more and more PDB entries, we actually have open access journals in PubMed Central.
Yes, as I said, we have actually started working on full text. I didn't present the results here because we're still analyzing them. Having an open access article, unfortunately, is not sufficient for enabling text mining. The current rules in PubMed Central don't allow you to access anything beyond what they call the open access subset, which is essentially a subset of, it's very small, it's less than 200,000 articles right now. So even though they may be open access, we may not in fact have access to process them computationally.
Yeah, I know those problems, but for many of the PDB IDs, there are still PubMed Central articles which are part of a public subset.
Yes,
I wanted to ask you if basically, if that gives you actually, if you have articles where the license is okay to use and all that's fine, if you actually get more value out of a full text of a text body.
So far, we haven't been able to show that because you have more noise in the full text. The advantage of the abstract, of course, is that it focuses in on the core results of the paper. So, as I said, we're still not done analyzing the results for full text. So, but the preliminary analysis suggests that actually there's some noise we have to deal with, and so moving to this kind of relation extraction is probably required to deal with that.
And
I think we're going to have to have time for one more quick question. Did you have a very quick follow-up, or that's okay?
Okay, a quick one. How do you control for negative information as well as uncertain information?
So we don't at all at the moment. As I said, we're taking the most, I would almost call it brain-dead hypothesis, which is that any mention is meaningful. Now, the negative information, of course, could be problematic, but we've shown pretty clearly that actually it's not affecting this particular case. But of course, that might be precisely because these are the primary references for the PDB structures. So we're getting some sort of, positive value out of the fact that these are already, validated. These are the most important. This is the most important source of information about those protein structures. So probably, if it's a negative context, it's not going to be in that primary reference. It's probably in work that comes later.
And uncertain, where maybe as a part of discussion in the abstract, they mentioned that it might be related to a functionality.
Brain-dead text mining, okay? I said nothing complicated. And that's not to say that we don't need to deal with all of those cases in order to improve accuracy. And that is, in fact, what we intend to do. But this work reflects just simple matching of those very simple expressions.
Thank you.
Is it really? Do you have a quick question?
I believe it is quick, yeah. Karin, thank you. You mentioned that you are doing function prediction toward the end, but it seems to me that you are just saying whether a residue has a function or not. I don't see where are you predicting the actual protein function, as you said, toward the end.
Well, so remember that I defined function physically. So we're equating here function with functional importance in the structure. So we're not predicting what kind of function is happening at that location, but what we're saying is that that particular site on the protein is important for modulating the behavior of the protein. So it's function in the physical sense.
Of a residue, but not of a protein.
Well, I'm not sure there's a distinction. So, I mean, the protein function is determined by the particular residues that are
Yeah, the thing is that usually when we say protein function prediction, like there was in CAFA, You want to know what is the function of the protein, like whether it's an enzyme.
So
or
we're defining protein function physically, and so these are all going to be catalytic sites, ligand binding sites, and that's not about the sort of downstream implications of what happens at those sites. This is a physical definition, and it's important for, as I said, the drug discovery context.
Okay, well, it's time for lunch, everybody, so thank you very much for this session, and we'll see you in the afternoon. So thank you again, Karin.